So for any pharmaceutical compound or a therapy, so you always have to worry about what is known as a therapeutic window. So therapeutic window is defined as how good is your molecule in terms of efficacy. So that is whatever it is supposed to be doing, and versus how bad it is when it gets into the system, that is called toxicity.
Yeah.
And so there is a ratio between the efficacy versus the toxicity. In small molecule chemotherapeutic compound, because of the hydrophobic nature of this molecule, so once you administer it into human patients, so it simply can diffuse into this thing. So in addition to reaching the target of interest, it also gets into variety of other non-specific organs also.
Yeah.
And so that results in quite a bit of a toxicity associated with the small molecule compound. Whereas with the antibody drug conjugate, antibodies are specifically selected against the target antigen, which are overexpressed in cancer and which normally do not show quite a bit of an expression in the normal tissues. And so because of that, so you have the non-specific toxicity due to the antibody drug conjugate going into some other organs other than the tumor site is very minimal, and so that's where you have an advantage over small molecules in terms of the toxicity. And so that is the reason why, compared to the small molecule compound, antibody drug conjugates are supposed to have a much better therapeutic window.
Okay, that makes sense. What are the, you said that on cancer cells, there are certain proteins or markers that are heavily upregulated or expressed greatly that are what presumably these ADCs are targeting.
Yeah.
Could you give us information as examples what these antigens are that these antibodies are targeting?
So the antibodies are usually targeted against the tumor-specific antigens. So usually under cancer condition, some proteins are overexpressed on the cell surface, and some of the functions are known, and some of the functions for other proteins are not known. But usually you do see some proteins that get overexpressed in this thing, and the level of those proteins compared to the normal tissue is much higher in tumor this thing, and in many cases it may not be present in normal tissues but specifically shows up in the tumor this thing. So the antibodies are carefully selected to have that specificity toward the tumor, and so that is one of the process one has to worry about when you are doing antibody drug conjugate, how you choose an antibody. And so you have to choose an antibody against an antigen which is overexpressed on the tumor, and it should be present in a fairly higher amount compared to the normal tissues. And the second thing is, so these antibodies, the prevailing hypothesis in antibody drug conjugate is it works due to the internalization of this antibody drug conjugate into the cell. So that means upon binding to the antigen, the antibody gets internalized due to receptor mediated endocytosis, and so it goes through these endosomal and lysosomal vesicle pathway. And so, and in the lysosome the endosomal there is a lower pH. So because of that lower pH, so you could use that system to release your cytotoxic drug that is associated with the antibody drug conjugate. And so that's so one of the specificity that one has to consider when dealing with antibody drug conjugate is, so it's not only the expression of the protein specifically on the tumor tissue, it also should be able to internalize the antibody upon binding to the receptor.
Okay, so you have to choose an antibody specific to a receptor that is able to induce receptor-mediated endocytosis. Otherwise, there's no point. So,
that's
okay.
the thing. Although there are many instances where non-internalizing antibodies are also being used, but the prevailing hypothesis right now is the internalization is one of the main criteria that is needed for the successful development of antibody-drug conjugate. But in future, it may change.
Okay. But as is then, according to that prevailing hypothesis, that drastically limits the number of possible antibodies and makes the selection process probably very difficult, correct?
Yeah, so that means that you really have to choose an antibody that gets internalized by these receptors for antibody-drug conjugate therapy.
That probably makes the process very costly, right? The development of these drugs?
No, it's a simple, the cost of making antibody-drug conjugate essentially is higher than the antibody by itself, because so you have to make cytotoxic drug also. So that itself is another process which requires quite a bit of investment this thing. And then you have to take an antibody and the cytotoxic drug and you combine them covalently, so you have to link them covalently, and so that process itself is not a straightforward process, because antibody is a hydrophilic compound, whereas the cytotoxic drugs are hydrophobic compounds, and so you have to make these hydrophilic and hydrophobic come together and stay in solution so that you could deliver as a therapy to the patient. And so that is a challenge. How you make antibody-drug conjugate is technically a challenging issue.
Thank you very much.